Good morning. May I have your attention, please? It's 9 o'clock, and I hope you had a comfortable night. Before I hand over the session to Dr. Agarwal and Dr. Kang, I'd like to make a few comments and some notice. In English dictionary, a new word has been coined called fobbing. Fobbing means phone plus snubbing. And if somebody is speaking and if you use your cell phones, you are essentially fobbing. And I yesterday received many complaints of many people talking in the hall while the lectures were going on. So may I request you that kindly avoid fobbing, kindly put off your cell phones. If you must take a phone call, please go out so that the others are not disturbed. And I'm compelled to say so because I got about 10 complaints yesterday and they wanted me to do something about it. I would like your inputs for the new symposiums next year. You have heard the kind of symposiums we had. What could be the new themes which will look at the future of Indian science in various aspects? So I will request you to think about potential new areas that we could cover in the next year's symposium. Your inputs to any of the talks in form of comment or an additional write-up are welcome, and kindly send this to respective conveners and copy to me so that we'll follow it up in the books that will come from this. Finally, we have in the afternoon from 2 onwards we'll induct the new fellows and give the awards. So between 2 to 3 we'll have this function. Then we'll have a tea break, and after that there will be a meeting of the fellows only in this hall. So kindly keep it. And once again I will request you to remember that like CSR, we have to think in terms of ASR, the academy's social responsibility. So I urge you to give me ideas that we can take forward. Thank you, and I welcome Dr. Gagandeep Kang and Dr. Anurag Agarwal to take this symposium. And I'll also like to once again applaud Dr. Kang for being elected to the fellowship of the Royal Society. Please give her a big hand. I'm happy that in spite of her busy schedule, she is here. And she looked much younger than I thought she was. For the achievement she has, you see her CV. So I compliment you also, madam. And I think you are the best person to talk about future of healthcare. Thank you so much.
On behalf of Dr. Kang, looking younger and walking up over here, and myself, I thank Dr. Singhi for this opportunity to talk about the future of healthcare, and thank the entire NIO team, his sister lab, for the wonderful arrangements over here. I think the topic of future of health is close to almost everybody's heart, especially looking at the average age of women. And it's something that is being very dynamically shaped by a number of macro trends that currently exist. The way we have envisioned this particular flow today is we start with things that are of absolutely proven benefit. Dr. Kang speaking on vaccines and where they might be going, to things that are of not so proven but high potential, which include genomics, artificial intelligence, which are tremendously going to shape the health and are heavily tech-related. But to bring sense to it all, because ultimately we all want a type of a future. We all want healthcare to be better. But future is just the future. It does not necessarily need to be better or worse. So what are the things to think about, what are the lessons to learn from yesterday, which will tell us regarding the choices we need to make going forward? And for that we have Professor Srinath Reddy, President of the Public Health Foundation of India. And in the end, we talked about CSR, ESR. There could be something in the middle. As scientists, we have responsibility not to just talk about the future, but to invent it, to create it, and perhaps to even form the corporates that will lead to this future. And Dr. Rajesh Bhokle will speak on that. I am very pleased that we have a very distinguished panel. I mentioned Dr. Kang, and we also have Dr. Manish Gupta from Google Research India, who will be speaking on the parts about AI, who better than him. So we will not waste any more time. We will start with Dr. Kang as the first speaker. speaking about the futures of vaccines and where we could go with that, from infections all the way to cancer. Thank you.
So thank you all for being here so early in the morning, and thank you for the very warm introduction and the welcome. One of the things about trying to get a lot done is to delegate as much as possible, and when you have enthusiastic volunteers like Anurag, that makes the job that much simpler. So what I'm going to be talking to you about is vaccines, where we are and where we think we are going. And given what has happened in the last 20 years, as you will see, I'm not so sure that I'm particularly good at predicting the pace at which the future moves. So if we look at what the world was like without vaccines, smallpox in the 1700s killed about 400,000 people every year. This is killed, not maimed, left blinded, and otherwise disabled people. So we all know the history of the smallpox vaccine, and Edward Jenner's seminal work in 1796, doing experiments that would not be ethically permissible today, actually resulted in what was the first vaccine. If we look at the timelines, 1796 was when proof of principle was established, and very rapidly for that time, given how important this vaccine was, it was scaled up around the world. However, a concerted effort to use this vaccine as a public health tool and potentially eradicate the disease actually came about only in the late 1900s. And starting from 1959, in a period of approximately 20 years, with countries working together under the guidance of the WHO and people from the Centers for Disease Control and Prevention, we were able to eradicate smallpox. Now, that is a disease that it was critically important. So far, it is the only human disease that we have eradicated. But we have eliminated and controlled many other diseases. If you look at the examples here, this is measles. You can see what happens once the vaccine is introduced. And that is for a disease that was endemic, that had been present in the country for a long period of time. Vaccine is introduced and you get control. On the other side, you have the example of meningococcal disease in New Zealand, where there was a certain level of meningococcal disease, and then it started spreading very rapidly in adolescents and college students. And they made a vaccine which is no longer available now, and that drove the epidemic down. So what do vaccines do? Vaccines are estimated to prevent about 2 to 3 million deaths a year. That's actually 5 deaths being prevented every minute. And this is counting only deaths, not the burden of disease that is alleviated. But if we look at what has been done with vaccines, starting from Jenner, going through Pasteur, going to Maurice Hilleman, who's probably the person who made the most vaccines in the world, the essential principles of vaccine development have been exactly the same. You know what the organism is, you isolate it, you inactivate or attenuate it, and then you inject it into people and wait for them to make an immune response that is protective. So up to the 1950s, this was the paradigm that was followed, and it resulted in a number of very successful vaccines that were made using these empirical approaches. But since 1950, we've had every few years new technologies emerging that have changed our ability to make vaccines. So if we look here, in 1950, cell culture was established. That resulted in the oral polio vaccine being made, and vaccines for a number of other viruses. The vaccine that we hype so much in India, the rotavirus vaccine, is actually a technology that was developed in the 1950s because you grow the vaccine in cell culture. If you look at 1980, recombinant DNA gave us the ability to make a hepatitis B vaccine that was not based on getting the virus out of the serum of infected patients. It also led to acellular pertussis, the older version of the HPV vaccine. In 1990, we had conjugation technologies coming up, and this now allowed us to make an immune response where you could take what was a polysaccharide antigen and produce a durable immune response because of conjugating the polysaccharide to a protein. And in 2010, we had reverse vaccinology, being able to use genetic information to develop vaccines. So since then, we've had an explosion of other new technologies, structural vaccinology, the ability to make DNA and RNA vaccines. We've had advances in adjuvants. We've had advances in our ability to interrogate the human immune response and design better antigens based on that. So what I'm going to do is take you through very quickly a few of these approaches. If you look at reverse vaccinology, the first vaccine that was made based on that was licensed in 2012 by the European Medicines Agency. This was the meningococcal B vaccine known as Bexsero, made by GSK. Essentially what they did was sequence Neisseria meningitidis and identify what might be potential antigens that would generate a protective immune response. They synthesized those, they tested them in animal models, chose the best ones, and then went into early phase human trials. The advantage of this approach was that you could do the first points up to the selection of the best antigen in as little as 1 year. So that allows us to change how vaccinology is done, because traditionally vaccines have taken 20 to 30 years to develop. Now, using reverse vaccinology, we have a number of targets. MEN-B was the first genome-derived vaccine, but now we are looking at what else do we need to be able to make vaccines for quickly. And we have some examples of success with B streptococcus, and we are also looking at a better plague vaccine using this approach. There is a lot of ongoing work on antibiotic-resistant organisms as well. Now, looking at structural vaccinology, those of you who follow the history of vaccines, one of the disasters in vaccines was the respiratory syncytial virus vaccine that was made in the late 1950s. What happened was children were given this vaccine and they had essentially a cytokine storm and died because of the RSV vaccine. So after that, people, one, did not understand why that had happened, and it's only recently that we have found out that it was because the actual protective epitopes for RSV are a pre-fusion form of the F-glycoprotein, and this is only transiently expressed in infection. So what researchers at the National Institute of Health have done is not only identified that this is a protein that is important, they've also used engineering techniques of structural vaccinology to make an antigen that is much more stable, a much more stable form of the pre-fusion F protein, and made it so that it induces a durable immune response. So we now have an RSV vaccine that is rationally designed and is in trials. Similar approaches have also been taken to HIV with somewhat less success than with RSV, but essentially it's been around looking at what the viral mechanisms of immune evasion are and seeing what can be done in terms of engineering the structure of antigens to be able to overcome those limitations in HIV. Now, there are other approaches as well, and these are approaches, DNA and RNA approaches are being used to take vaccines beyond infectious diseases to cancers as well. So I'm going to give you examples largely of two companies. One is Inovio, and Inovio focuses on DNA vaccines. They make DNA vaccines for both infectious diseases and cancer. What they do here is identify either the target antigen of a tumor or an infectious agent, usually a virus. They sequence that, they use that to be able to make immunogens. They take those immunogens and put them in a plasmid. That is then made into an immunotherapy that is delivered through a machine that they have developed called the Selectra, and that induces an immune response. Shown at the bottom here, and I think the slides are not projecting particularly well, but if you look at the brown color on the slide down there, this is immunohistochemistry showing the presence of human papillomavirus in intraepithelial neoplasia in the cervix. What you can see is pre-treatment. There is a lot of brown there, and then if you look 36 weeks later, the HPV has practically disappeared. This is also paralleled by the presence of CD8 T cells, which are not there when in the pre-immunization sample, and what you can see as dark lines there demonstrates their presence and persistence in the tissue. So you see patients who recover from cervical intraepithelial neoplasia. So this is potentially a therapeutic vaccine. But these vaccines can also be used for infectious diseases, and there are good examples for both Zika and MERS. The MERS vaccine has gone into phase 1 human trials, but the levels of protection that have been seen with both of these vaccines are high. Now, if we look at RNA vaccines, this is essentially mRNA being put into the body, resulting in the formation of a protein. There are many different mRNA delivery systems, and a lot of the processing that happens depends on what you are looking at. It may be the protein by itself, or it may be the protein that is presented in the context of histocompatibility antigens. So there are a number of mRNA vaccines which are in trials for infectious diseases and for cancers, and a number of companies that have been established are looking at both infectious diseases and cancers as targets. Two companies, CureVac and Moderna, are probably leaders in the field and have multiple products that are now in clinical trials. Now, if we look at adjuvants, adjuvants have tremendous potential, but so far we haven't had a lot of them. Alum is the adjuvant that is in most of the vaccines that we use now. But GSK has a suite of adjuvants that are essentially squalene and water or squalene and oil emulsions that can change how poor immunogens perform. So this is an example of influenza vaccine. So you get about 43% antibody response in children in the absence of adjuvant, and that goes up to about 86% with this particular adjuvant, which is MF59. If you look at the licensed adjuvants, there are very few. The ones shown up on top, alum, MF59, ASO3, ASO4, and adjuvants have now become very hot property in the vaccine field. There are a number of others that are in development at the moment. The problem with developing vaccines now is literally the amount of time it takes and how little we understand how vaccines work. It's frequently said that the easy vaccines have all been made, and what's left for us are difficult targets. So we have problems because we don't understand how vaccines protect, and we have very poor ability to select what might be good vaccine candidates. So one of the things people have been thinking about is how do you avoid the idea of using too many animal models and get your information directly from humans. These are called exploratory clinical studies done in people. These are intended to generate or test a hypothesis very early so that you can decide on vaccine product development. So usually these are in small numbers of people up to phase 1 trials, and these also fit under a definition of what is called experimental medicine. So what is the traditional pathway for developing vaccines? It's discovery, preclinical development, phase 1 trials, phase 2 trials, succeed in that, and then go on to phase 3 trials. But if you use an experimental medicine approach, what you do is go from preclinical proof of concept to a clinical proof of concept, and then use that to inform exploration so that you can do studies that will de-risk your vaccine development. So if we look at rational vaccinology, I'll just take you quickly through this. You start with healthy volunteers, you use controlled human infection models, use those infected people to decide what are the antibodies that are relevant for protection, and then do antibody optimization and validation in the human challenge model. This allows you to do better epitope identification. You can make viruses or vaccines, make better immunogens, and those can be also taken iteratively through a fit-for-purpose animal model. And this now allows you to take that immunogen into actual early phase vaccine trials. So current pathways for vaccination follow a very, very traditional system where you have the antigen and you do preclinical and then a range of clinical studies resulting in the registration of the product. However, there are a lot of discussions around regulatory strategies that will allow us to make vaccines faster. Can we actually use animal studies in certain situations? For example, in Ebola, how would you test a vaccine? Currently, we've managed to make it through a phase 3 trial for one vaccine, but is that going to be something that will be feasible for future vaccine candidates? So in outbreaks, could you use animal studies? Could you use human challenge? Could you license early and then have a requirement for later phase studies? So in addition to the science advancing, the regulation advances, and the goal really is to have vaccines for every age and stage. So far we have focused mainly on children, but there is a huge need to be thinking about pregnant women, about adolescents, adults, and the elderly. Some vaccines are available, other vaccines are still in our future. The idea of vaccines is prevention. In the case of cancer vaccines, both prevention and therapy. And we have a number of vaccines that fulfill the goals of public health, which is how do you prevent disease in the maximum number of people for the lowest cost possible. So you could think about vaccines as being a poverty alleviation tool as well as one that promotes equity. So with that, thank you very much for your attention.